These are my disclosures. I can't say this talk would be the same level and complexity of the previous one, but it will be light to hear, so please bear with us for another half an hour. We will touch upon outcome measures, what is needed to measure a curative therapy, and gene therapy may be a cure for hemophilia, as we have heard many times in many different views these days. Efficacy, safety, nothing new. Cost effectiveness, health technology assessment. So we'll move a bit after the regulatory approval, value proposition, and some issues around measuring outcomes. Now, if you think of the entire cycle of development of a new drug, you start with research and development, early clinical testing, and then you move into true late clinical testing. You get through the regulatory approval. I won't add anything to that step. But then you also have HTA. You have to check or assess the cost effectiveness of a new treatment. And finally, you have to find a payment model to make the treatment available and sustainable. Now, if we look at this step, I will say nothing on this phase. It's not my field. I'm a clinical epidemiologist. And also, there's nothing really I could imagine more than we have heard already during this meeting. I will move into late clinical testing when you are reasonably convinced that you have something valuable in hand, and then you may compare. Now, I'm using a classical approach where you would randomize patients. We likely will not need and not have randomized trial in gene therapy for hemophilia, but you will anyways need to compare. You have heard across all the entire previous lecture. And you will have someone who received the experimental treatment, gene therapy in this case, and you will compare with the standard of care, whatever it is, depending when the trial will be run. Can be prophylaxis with factor concentrate, can be mimetics, or can be an historical control. But you are comparing two groups. And you observe them over time, and we will need to discuss how long that observation needs to happen. And during that time, you measure how many good or bad outcome happen. And of course, you can have good and bad as calling as bleeds is bad, no bleed is good, or high measurable factor level is good, and factor level below a given threshold is bad. Doesn't really matter what the outcome is, but what you are doing, you are counting in the different groups, who's bleeding, who's not, or in the same patient before and after gene therapy, bleeds or factor level, and you are comparing. And this is the way the late clinical testing happen. Now, who provides input into the choice of outcome? If you think not of gene therapy, forget for a second, I know it's hard, but don't focus for a second on gene therapy. Think of the previous treatment. You would have sponsor and researcher proposing trials and proposing outcomes for the trials. And patient input. Of course, patient input has been growing over time. We didn't have that much patient input a few years back, but now with PCORI and with all the focus, we are very appropriately posing on patient-centered research. Patients are asked more and more often about what they think make the important measurement for a new treatment to show to be effective on. And of course, here is where FDA, EMA, or whatever regulators is ruling a specific setting will have a say. So up to here, mostly, is efficacy and safety. It was said clearly by Dr. Iorio. They don't look at FDA, at cost, they don't look at access. It's mostly all revolving around efficacy and safety. But if you move further on and you go to health technology assessment and you look at what happened later, well, you don't do trial for HTA. It's not that after regulatory approval, then you start doing another trial. The decision made at the HTA and market access level are based on the same trial, with one difference, that usually at the HTA level, you don't look at one trial, but you look at all the trial that you have in the field. And you try to get some judgment about what you know overall about the treatment. And again, there's not only hemophilia that we need to care for at the HTA level, and you start looking at where is more appropriate to spend public money among the different treatment available for one disease or for many diseases. And of course, if you look at the trial, well, it's still patient and sponsor that have one trial at a time set together and decided which outcome were worth. And the trial have been measured across, the outcome have been measured across trials in a more or less consistent way. So ideally, you look at the same things across different trials. The problem is that for many reasons, one is the creativity of the investigators, and the second one is the competitive aspect of medical research, where in the end you want to know what is good, but also what is better than what your competitor produces. You start being very creative with outcomes, and you may not have a lot of evidence that let you compare different treatment. And if you look at this from your narrow perspective of the one who did study 1 or is interested in outcome 3A, you may see that is a winning point. But if you look at it from a HTA perspective, this might be a place where everyone loses, because you cannot really show what the result is. And you may have a much better game if you have something that you can compare across trial, and you might not be able to show differences between different approaches, but you will be able to show how powerful that approach as a general approach is. This is not a problem only for hemophilia. For example, we have cited already HIV and AIDS, and there was an extreme variety of outcome. Please note these names, Saldana, and also note Williamson. They did this study last year showing that the variability of outcome in HIV trial has been such that it was difficult to draw a conclusion. Now, there's another difference when it moves to HTA and market access. You are not only assessing efficacy, you are also assessing value. When it comes to paying for a treatment, you are interested in what you gain for the bucks you put into treatment. And so this is cost effectiveness, and is also what can be the premium value for a premium effect. If the treatment gives you something more in terms of efficacy, in terms of safety, but also in terms of all the other outcomes that are relevant to patient, and being relevant to patient, are relevant to health technology assessor, and are relevant to payers, you may need to do other measures to show what the value of a treatment is. And it's not that we have to, again, reinvent the wheel. There is an established way of measuring value. And there are actually more than one. The one I prefer is the one proposed by Porter and published now about 10 years ago, and applied to different diseases. Now, we have a nice application, is a proposal of application of a Porter approach to hemophilia, recently published by Brian O'Mahony and colleagues. Jerry Dolan was involved, Diane Nugent. And the Porter approach to value in hemophilia revolves around the Porter approach applied to hemophilia revolves around the three main aspects of assessing value, which are the level of health you achieve or retain, the process of recovery, and the sustainability. And if we decline these specific aspects to hemophilia, the health status, the achievement or retainment will be mostly function, activity, joint status. Nothing new. It's just a matter of measuring it properly and looking at what is the value that this brings to a treatment. Bleed severity, more than not count. Pain. Pain as a symptom, pain as a condition, pain as interference. Health-related quality of life, and doesn't refer to any specific tool, but refers to a specific life experience. And then, under process of recovery, you may have the time it takes after a bleed to get back to normal, and all what you lose in that period. Missed day of work, missed day of school, and inhibitor pathogens, orthopedic intervention, all of these belongs to process of recovery. Sustainability of health. Bleeding frequency, joint preservation can count here as they do in achieving or retaining health status. And what's the impact on lifelong productivity, good health? You may start shaking your head and saying, are you sure you can measure all of these? Well, first of all, it's not me proposing to measure this. It's a Porter, and is a requirement that more and more often comes from health technology assessor when FDA or EMA had approved the treatment, but then you know how long it can take to get something on the market, because this is what is looked at that stage. Now, if you think of assessing value, not all outcomes provide a contribution. So if you think of death, is a clinical outcome, is not patient-reported, is objective, is patient relevant? Certainly. Is HTA relevant? Yes, because it impacts productivity and it has consequences. Now, if you think of factor level, at the opposite end of the spectrum, is clinical? Yes, is a clinical outcome. Is patient-reported? No, is measured in a lab. Is objective? Yes, we may disagree on what's the best method. We may disagree on how precise the method is, but is, in a way, is objective. If you insist measuring, you get to some precision. Is patient relevant? Is not. Now, you may have some doubt, and for example, looking at the biomarine study, where both the high and low dose got 0 bleeds, the quality of life improved much better, much more in the group treated with the high dose, and one may suggest that knowing that your factor level is higher has a relevance for the patient. So one might challenge my assertion that is not patient relevant, but if you had asked this question 5 years ago, many patients would have told you, I don't really care where my level is, provided I'm not impaired in having a normal life. Now, is HTA relevant? Well, potentially not. I'm not sure how you would value being at 25 or 30. What's the additional value being 5% higher? Bleeds, they are clinical, yes. Are patient-reported? Yes, they are patient-reported. We can try to make them objective, but in the vast majority of cases, these are patient-reported events. Are they objective? I don't know, and I don't think it's that worth discussing for reason I will show you later on. Are patient-relevant? Again, we may agree or not. I recall many symposia and many talks where the target of 0 bleeds was not considered the most important outcome by patient, who were saying, I don't really care if I have 1 or 2 bleeds, more or less, provided I can enjoy a normal life. Are HTA relevant? Yes, they are, because there is a cost associated with their treatment. Pain? Pain shows up very well on this table, is clinical, is patient-reported, is objective. We all know that scales may vary and you may cheat on a scale, but there is a measure if you respond honestly to measure pain. is very relevant to patient, is very relevant to HTA. If you ask them, pain is something for which society is always willing to pay. Quality of life is not clinical. Doctors tend to say this is not something we are really excited about measuring, not that we don't care for our patient quality of life, but it's not something that is usually not the doctor proposing to put it as an outcome. Is patient reported? Yes. Is objective? Yes. Is patient relevant? Is HTA relevant? Convenience is down there. I'll stop going over the column just to tell you that not all outcomes are the same. Now, if I look at what are patient relevant and what would be good outcome for clinical trial and regulatory, could be possibly the first three. The last one, I'm not sure. If you look at HTA, maybe these are the more important. So different stages of the process may focus on different outcomes. Why don't we focus on those that have something to tell to everyone? Why don't we try to find the minimum core of outcome? Then we can collect all the rest if we like. But if you want to be sure, we shorten up the arch from research development to availability on the market. If we look at the entire spectrum, we would need to be sure to start collecting as early as possible those outcomes that are critical for all the phases, those that are of interest to any one of the involved stakeholders, so that we don't find ourselves after FDA approval to say, oh, this was one of the known unknowns. We knew we needed outcome important for establishing value. We forgot to measure them, so we have to start over again. Now, if you look at, and you may say, well, why are you wasting our time? Well, because that's what happened. I have reviewed what was the application to Curative, the health technology assessment in Canada, for drug application in the field of rare diseases. So 63 of 400 in 10 years were for rare diseases. Most had at least one small trial. So that may not be the case for gene therapy, but still, small trials, 190 maybe look big in the field of gene therapy, but there were trials as small as 20. Treatment cost was on average $200,000 Canadian dollar, so it's less in US, of course, but up to 1 million. So not things completely off ballpark for us. And there was one withdrawn. Only 8% was approved directly. Many were approved with restriction, and slightly less than half was rejected. And you may wonder how many were rejected on cost. Very, very few. The rejection is almost never on cost for rare diseases. It's because there was no evidence or no effect. Now, we are all happy if we don't pay for something that has no effect, provided we have done a proper job in trying to show the efficacy of that treatment. But it's really a pity getting here and being told, I'm sorry, you thought you had generated the right evidence. Even FDA thought that was enough to approve it, but we can't pay for it because we have no way to assess what are we paying for. And this is a large chunk of evitable failures. One way of reducing this chance of failure is to agree since the beginning what you are going to measure. And I asked you before to focus on Williamson. Now, for the transitive property, focus on Tunis. In the same group, I don't know if you know Doug Altman, Mike Clark. These people made a lot of the history of clinical trial design, and they have come up as a systematic reviewer. This all stemmed out of Cochrane and the attempt of making it easier to some result across different studies. They came up with an approach to agree before the trial are conducted on what is important to measure. And they suggest a few standards. I'll showing you what you need to know. Out of the 13 standards, they want to listen. They propose that outcomes are selected by listening to healthcare professionals, and that's okay, with experience with patient in that condition, so not just researcher, treaters, patient with the condition, and the initial list of outcomes should consider patient and treater reviews and should be clearly spelled out. We should make any effort to be sure that we understand each other. We have done this. We have done it for hemophilia. We have done it a bit in a rush, in less than 1 year, because trials are starting, as you know, and we wanted to be sure we were not wasting time, resources, and patient participation to trial. The Tunis, Sean Tunis, that I pointed you before as being one of those issues in the core study, was participant, actually was one of the co-PI in this study. That's myself and Glenn Pierce was in the group as well. You'll see the other there. This paper is published at the beginning of this week, on the 21st of May. You can have a look. The three institutions who were steering this process is McMaster University, my own, Mark Skinner on behalf of NHF, and Sean Tunis for the Green Park Collaborative, brought in the experience in the organization in setting core set. But who did the process? Were clinician, government agency, sponsor, payers, and HTA, gene therapy researcher, method expert, patient and patient advocates, payers. And you see, they come from different perspectives, different colors. What we did, we mixed them up as best as we could. We shake the bowl many times to try to have them talking to each other so that they could agree on a common set of outcomes. We went through a Delphi process as suggested by the COMET initiative guidance. We started with almost 60 outcomes, and we eliminated some. Some new were suggested. If you start asking these kind of people, they will never stop suggesting something new. But they agreed that 36 was a good point where we could have a conversation. We continued voting. The Delphi process is a process where you continue to select the most important, and you don't know who else is voting what, but you see that there's some convergence. We used a rule that we retained all outcomes where there was more than 70% of participants to agree, but we wanted to make sure that we were respecting and giving the due consideration to the patient preference and opinion. So whenever the patient component of the group was in disagreement with the others, but was in agreement between them, and the vote independently given by the patient representative was above 70, we put that outcome back. Then if it was kicked off again, was off. But at least we signaled the other stakeholders that patient felt they were important. To make a long story short, at round 2, we were already able to select 3 outcomes. For efficacy. For safety, we found ourselves particularly discussing with regulators. We were told, listen, you may select or not safety outcome. We are not going to listen too much to you. Safety outcome need to be recorded anyways. Why don't you focus on efficacy? Which is what we did. And so in the last round that we had after an in-person meeting, and it was an interesting meeting that we had in Baltimore, with, you'll see the list of participants is quite impressive, and you can read all the name on the publication. We got finally to cut down from 60 to 6 outcomes, and not to keep you on the hook for too long. These were the safety outcome, and the safety outcome, I don't think I have anything to add. I just leave you 30 seconds if you can still read after a day looking at the screen. These are the safety outcome: liver toxicity, short-term immune response to gene, clots, other secondary disorder development, and so on. But we were able to focus on efficacy outcome. On the outcome are organized by domain, if you follow the COMET approach. In the clinical physiological, we have frequency of bleeds. We call these a legacy outcome. We don't think this is the more important, and actually during that meeting, we had FDA present, and was Peter Marks, heading the hematology department. He was quite vocal in saying that he wasn't sure that ABR was to make it as an important outcome. He made it clear that FDA is no one person. FDA is an impalpable entity out of which you get decision, and you'll never know who said in the end what. But he was very, I'm not FDA regulated, so I'm able to overspeak. Factor activity level was actually pointed out as potentially more interesting for a treatment able to go to 50 or more % of factor VIII, and is a strong argument. And we, by the way, we have lived by approving all the first, second, and third generation recombinant just based on bioequivalence with plasma-derived for the factor level activity they were able to produce. So this would be just on going back to origin, not creating anything new. Duration of expression, these are the three clinical. Pain, discomfort was a domain per se. Chronic pain and interference was the one that got more agreement across the board. Utilization, so direct cost is what the payer are interested in particular, and emotional functioning or mental health. We'll need to come back on this in a second. Now, it's not only that you have to define what the outcomes are, you may need to provide detail. It's not only health-related quality of life, which instrument you use, are you measuring the change over time, are you looking at the patient crossing a threshold, when are you doing the measurement? All these detail, I pointed before to the name of Saldana. We will be working on giving instruction, suggestion about how to do the measure for the outcome we have selected. Just looking at quality of life, you may see that before and after you go from 27 to 35, and if someone tells you that with that specific instrument, 6 is the difference that a patient can perceive as making a measurable difference, you might say, well, yes, the difference is more than the average difference, but you still may have a lot of variability. How you handle this? Well, you would need to look at all patients and then see who went what. Some patients had an improvement above 6, some had an improvement but was not 6, and some other actually had a worsening. So a different way, instead of saying when 27 to 35, would be 6 in 10 patients had an improvement above the threshold. All these details can make the same outcome, in this case quality of life, meaningful or not for the health technology assessment. And I would also humbly say for the patient, it's easier for me to tell a patient 6 in 10 patients get a measurable improvement in quality of life than telling someone your quality of life goes from 27 to 35. Thank you. I would like to be at 100 if that's where the scale goes. Now, last example I'm showing you, this is not about gene therapy, this is about emicizumab. Many improvement, don't take me wrong, I think is a powerful treatment, but if you look at bleeding events, the way they are reported is appear to be reduced. Now, we have heard many times during this Congress that bleeding are subjective. Well, actually bleeding has been subjective forever. It's not that they became subjective now that we are assessing gene therapy. No, we didn't complain that bleeding were subjective when we were assessing long-acting or we were assessing previous treatment. The community need to have some historical memory. So the fact that something is subjective as a solution, if a trial like this where you had compared emicizumab versus on demand and you were counting bleeds, and we know that bleeding are subjective, you are on treatment and you may call a bleed and you are not on treatment, you call more bleeds, or you are on treatment, you jump on a trampoline and here you don't. Since the event per se is treated with bypassing in inhibitor patient or with factor VIII in non-inhibitor patient, would have been enough to blind here and give a placebo injection for the time of the trial, for knowing if this measure was reliable or not. Was completely ethical, is done in all fields. You don't put anyone at risk. You just tell, I don't know if you are on EMI or not, so be patient for 6 months, live as if and let's count the bleed. So there are opportunities. I'm not saying we have to blind gene therapy trial. I'm just saying there are ways of making all these outcomes measured in a way that is credible to every stakeholder, having the patient center and front, but having everyone else finding what they need. We will continue working on a way of defining the transformational impact of gene therapy. There is no tool available, and we will work as coreHEM group in developing one. I have used as my summary slide the list of outcomes we have identified in the coreHEM set. There will be ISTH recommendation, there will be WFH recommendation. This is the group of people we call together. Is a quite large group of people who already agrees. It doesn't harm having other recommendation. We think we have done a quite good job already. And on this one, which I'd like to leave up, so these are alphabetically ordered. If you look for someone, you can look on the slide, but I'll conclude here my talk. Thank you very much.